All attendees are in listen-only mode.
Good afternoon, everyone, and thank you for attending our fourth live webinar session of this year. My name is Marine Letertre. I'm currently working as a facilitator of the Metabolomics platform Corsair in the west of France, and I will be hosting the webinar today with Dr. Helena Legrand from McGill University in Canada on behalf of the Early Career Members Network of the Metabolomics Society. As you may already know, the webinar is free and will be recorded so you can freely access it on the Metabolomics Society website. Before to begin, I would like to invite you to follow us on our social media, Facebook, Twitter, and LinkedIn, and please do not hesitate to contact us at the email address indicated on the slide to give us your feedback about the EMN or any suggestion. With that said, I would like to thank you, our two speakers, for accepting to present their research during this session. We are glad to welcome Dr. Justin van der Hooft, who is an assistant professor at Wageningen University in the Netherlands. Justin obtained his PhD in 2012, during which he developed methods for systematic metabolite identification using both MS and NMR techniques. He then moved to Glasgow for a postdoctoral position, during which he worked on various projects, such as bacterial metabolomics. This position also gave him the opportunity to define the concept of MS2-LDA in collaboration with Dr. Simon Rogers. In 2017, he moved back to Wageningen to perform a joint postdoc with the group of Dr. Peter Dorestein and the group of Dr. Marnix Medema, focusing on combining genomes and metabolome mining, subject on which he is still progressing thanks to a 3-year grant which aims to develop computational tools to decompose complex metabolic mixtures in natural products. It's a pleasure to welcome him today, as he was a founding member of the EMN and he is on the board member of the Metabolomics Society since 2015. Following Justin's presentation, we are happy to welcome our first early career researcher, Dr. Madeleine Ernst, currently working at the Danish Center for Neonatal Screening at Staten Serum Institute, where she developed computational approaches and data analysis pipeline to explore metabolomic signatures of neonatal health and disease. Before that, Madeleine performed a postdoctoral research in Peter Dorestein group at the UCSD, where she worked on various data mining tools, such as GNPS and MS2-LDA. She obtained her PhD through a Marie Curie initial training in plant chemistry at the University of Copenhagen in Denmark. So thank you to our two speakers. Don't hesitate to send us questions through the chat box, and we will be able to relay them to our speaker at the end of the presentation. Just before to start, I would like to mention that our next webinar will be held on Thursday, 16th of July, during which two early career scientists, Dr. Pruva Kulkarni and Melanie Odenkirk, will be presenting their work. So with that, I would like to welcome Dr. Justin van der Hooft, that I'm going to make presenter, and we will be able to start.
Thank you. So, can you hear me?
Yes, we can hear you.
Good. And can you see my screen?
We cannot see your screen yet.
Okay. Then you still have to make me presenter.
Yes.
There we go. Okay. I think we're set up, right?
Yes. The floor is yours.
So, thanks so much for your kind introduction. And indeed, as you rightly pointed out, I've been a founding member of the EMN, and it's a real pleasure to be in the webinar series today that I, together with Eva, back then in Glasgow, set it up and started. And we were so happy that back then a number of metabolomics researchers were happy and eager enough to join the series and provide the webinars. And in the beginning, we had like 30 people and 40 people. and that number slowly increased over the years. And I'm really happy that today this is a still ongoing webinar series, and especially in these turbulent times with the coronavirus, it is really nice that we can share our research like this. So thank you so much for the organization and all the hard work to make it happen. So today I will share you some work on how we can decompose complex metabolite mixtures to substructure-based metabolomics workflows. And this is work that has been developed over the past years, and I will share with you mainly today the concept of MS2LDA substructure finding in LC-MS and MS data. So, oops. So first, I want to highlight, as I always do in my talks and webinars, that the work I show is obviously teamwork, right? It's a collaborative effort. It's a community effort these days to really gain as much information from metabolomics data as we can. So I want to highlight the Medema lab, headed by Marnix Medema, and yeah, over here. And I want to highlight the eSign. So and over here we see Simon Rogers, with whom I designed the concept of MS2LDA. So to start off, if you think about metabolomics, and we think about application this year for the third year in a row, it's very dry, and that's already asked questions about how can we maintain crop growth, and how can we make plants resilient against those different climates. And if you think about our food production, then lots of it is dependent on staple crops. And we can see examples here of foods that I think most of us eat on a nearly daily basis. So another question that is related to this, or another field, is not only do we want to grow these plants, but they also should be healthy. If we eat bananas, we expect that we get nutritional value out of them. And the impact of climate on how healthy our plants are is only a field that is just emerging now. For example, another field that is emerging is the plant microbiome field, where now we start to realize that plants are dependent on, or that many plants are dependent on microbiomes that form a layer around the roots and are even living inside the plant to sustain and also help to fight off pathogens. So now we know that plant growth and health and nutritional value are all very important topics. What is the relation with metabolomics? Well, many of these processes are heavily dependent on small molecules. And we think about, for example, resistance, then antimicrobial compounds, antibiotics, for example, can be produced by microorganisms, and they can help to fight off pathogens. In a similar fashion, plants also produce small molecules that help them to repel insects or other pathogens. Finally, if you think about health, and if you think about eating plant-based resources, then many of the small molecules, including sugary compounds, fatty acids, etc., are also playing a huge role in how in the nutritional value of the plants. So let me start by asking a relatively simple question. So that we know these metabolites, and we know these two metabolites. One is a polyketide, one is a peptide. Then we can start to ask questions like, where are they coming from? And if you are a if you are working with bacterial data, then you might realize, oh, they are most likely produced by bacteria. And if we know their genomes, we can start to predict which bacteria can produce which kind of molecules, and we can start to make the links. Of course, that doesn't answer questions like, what do they do when you find them in the soil of plants or in our gut, for example. But at least we know then the structure, we can know where they come from, and we can start to form hypotheses on why they are present or what they are doing. However, this question in practice is not as simple, right? Because normally we don't look at structures, but we look at spectra. In the talk today, we will mainly discuss mass spectrometry data. So that's an example here of a mass spectra. And then we see question marks all around, right? Because now we have a spectrum, We don't know yet the structure, so it's harder to find out who is producing it. Is it bacteria? Is it plant? Is it a human? And then it's even harder also to answer questions like, what is it doing? So therefore, my research vision is to really close the gap between what we can see in metabolomics, so the spectra, and what we can actually learn from it. So the biochemical and the chemical interpretation of the data. And today, a large role is there for pattern mining. And to illustrate that, this is a picture I took in the inside of a church. And this church has been built over many, many years in the last century. And if you look carefully, we can observe a lot of patterns, and patterns that are inspired by nature. If you look here, to me, it looks like a flower, and I'm pretty certain that that was one of the ideas behind it, right? And if you look more closely across this church, you will see patterns everywhere. So nature is full of patterns, and the human brain is really keen on identifying the patterns. So patterns can help us to simplify complex world, and that we will also do today with the metabolomics data. Talking about metabolomics, just to illustrate some typical metabolomics workflows that are around these days. So one could do a targeted metabolomics workflow where we start with what we know, so standard metabolites, and then we end up with, we can ask questions like, are they different between two groups? And we can also start to quantify them. The other way of doing metabolomics is untargeted metabolomics, where we start with a question more about the global profiling of samples. And we end up with a lot of data for where we still need to get the structural information out to start to answer the questions about the data. So instead of getting a direct comparison between molecules, we get a comparison between metabolite features that we still need to couple to structural information. Today's talk will be mainly focusing on how to get more out of this untargeted metabolomics workflows. Okay, so to decompose a complex mixture, it's important to get a good LC-MS and mass measurement. And that all starts with LC, right? Liquid chromatography. And very briefly, we do that to really separate out the components in your complex mixture to not bombard the mass spectrometer, but all of them in one go. And the concept of chromatography the word comes from the Greek word for color. And I don't know, I did it when I was young. I don't know if you did it, but then I challenge you to try it and circulate the picture on Twitter. So I took toilet paper and I put a black ink mark on it, and I put it in the water, and then I waited a few hours, and indeed, over time, the colors from the black ink, which is composed of various colors, would be separated out on the toilet paper. So kitchen roll paper should also work, by the way. So just make sure that the black dot doesn't end up in the water. That's important. So in real life, we don't have these colors and the ink spot, but we have the complex mixtures. So we throw them up on this kind of columns, and then they are part of this LC-MS setup, where in fact that is over here, the column, and it is the interface to coming from separating the samples, and then we arrive in the mass spectrometer where we can obtain our multidimensional data. So that is because we have both in retention time, we have an access, and also in the mass, in the MZ area, in the mass area. Then why do many labs now use high throughput? And overall, most methods cover a large amount of the metabolome. So an important and why do we do that? Well, to get additional structural information about the things we measure in the mass spec. And you can see here how we have ionized molecules. They arrive in the mass spec, which is now represented here by a black box doing several things. And we get out an MS1 spectrum. So here, these are the ionized metabolites. And then we can, within the mass spec, we can set up a method to actually select some of those and obtain a fragmentation spectra, which you can see over here, MSMS or MS2. And then we can start to do things with them. For example, match them to experimental data. We can match them to in silico spectra. I won't really cover those aspects today very much, but today we will cover the computational methods that are around now to actually start to get the structural information out of this MS2 spectra. To explain some of the concepts in a bit more detail, here we see a spectrum of histidine. And this MSMS spectrum in literature, you will also see terms like barcode and fingerprint. And the reason for that is that a lot of molecules have a unique fingerprint, a unique mass spectrum when you do the MSMS measurement. And here we can recognize mass fragments and neutral losses. And together they contain structural information about the fragmented molecule. For example, the fragments here, they correspond to this imidazole part of the histidine, and the neutral loss here, the yellow one, that is actually this carboxylic acid group that's chopped off the molecule. So even without knowing the structure, I can already start to hypothesize how the complete structure could look like. So the real challenge in the field is, especially in natural products and plant metabolomics and actually all kind of fields where we are challenged by or faced with complex mixtures, is that we have a really lot of molecules. And if you look out in nature, there's many examples of molecules that are found and identified, elucidated, identified, and actually used by us, hijacked by the humans to do a very good work for us. So, for example, as an antibiotic or an antifungal compound, but also insecticides, etc., etc. So many of those molecules also do things that they were not originally designed for by nature, and that's really interesting. However, they are part of a complex mixture, and the challenge is to, out of this mass spectral data that we can obtain from them, really couple the known structures, but especially the novel structures that we don't know yet, and that could be the next blockbuster antibiotic. So that is the real challenge. And the classical route of doing things is really to take this fragmentation data out of LC-MS data and then do kind of one by one matching to the databases that are there, the growing databases, but they grow relatively slowly, to see if you can find a match. And this typically works for 2 to 5% of the molecules in a complex mixture. But as you can see, that leaves most of the data actually unexplored. And I put down a number of libraries here and tools that you could use to do this approach. So also we have the GMPS libraries these days and mass banks, so there's many more. But overall, this route has an limitation. It doesn't use all the data. However, if you have the time and the dedication, then you can do a lot of nice things. These are examples of my bacterial work from Glasgow. So, for example, this E. coli metabolites, I used Magma, that's a tool designed by Lars Ritter, and I used that to help me annotate this molecule over here, arginic acid, and that was really nice, and that helped me to make confident annotation of this metabolite in the data. And not only that, I also elucidated novel molecules using the fragmentation data. So over here, we see on the right side, we see an example of a difference between two spectra. And I could link that to the presence or absence of this acetyl group. So depending on the spectrum, the acetyl group is moved position. So I could actually annotate these two isomeric compounds. Another example here on the left is a completely novel molecule that was by I think, as far as I could judge, never seen or reported before. Using these two fragments over here, the 71 and the 70, that I could relate back to this part of the molecule. And then I could start to build the rest using the other information that I had. However, this takes a lot of time and effort, and you actually want to do this in a more higher throughput. And that is one of the drives for improving these annotation workflows. And so we know now, I've shown you examples how MSMS information can be key for the elucidation. I've shown you how I think pattern mining can help us. We already started to see some patterns in the fragmentation data. Some fragments are similar, others are different. However, when I first looked at this LC-MS data, it was really messy and cloudy for me. I really had to go through the data one spectrum by one spectrum. And it took me nearly half a year to analyze one LC-MS file. That's a lot of time. So one of the motivations as well is to speed up the process a little bit and make also sure that we can integrate the various workflows that are around. So when we want to decode spectra, we actually want to reach the structural information that is in the spectra. And here I have made a schematic overview of what that means. So say this is a structure, and these are the fragments that we can expect in the mass spectrometer. Then this could be its spectra, right? And remember the terms, mass fragments and losses. So over here we see the blue part is responsible for these two fragments, and the brown part is responsible for the loss here. When we see this molecule here, we can visually observe that it's structurally very similar, right? Overall, it's almost the same. And when we make fragments, we can still observe that there are similarities. However, the final spectrum is already starting to appear a little bit different, right? We still see the blue peaks, but the brown loss is already grown a bit. So the challenge now, the challenge of the tools is to recognize what is similar between the structures behind the spectra, but still also kind of capture where they are different and how they are different. And that is what many tools these days try to do, and with increasing success. Today, I will highlight one of the tools, MS2LDA, that is focusing on finding the patterns that we can already start to see emerging here of the blue peaks, right? Same substructure across various molecules, but they correspond to the same fragment peaks in the fragmentation spectra. And this is a picture that I took near San Diego now 3 years ago. And I took it because I immediately saw, and I thought that was nice, there are two groups, right? We have the seals and the seagulls. And basically, that's kind of representing what I'm trying to do, or what I'm doing with metabolomics data. I want to find molecular families based on the spectral similarity, as I just showed you. And also, I want to extract the building blocks of metabolomics, right? The substructures. And the substructures are also part of my title. So that's what we will mainly focus on today. And then you can start to use this grouping to do all sorts of nice things, like propagating structural features and annotate chemical families. So I think the solution for the challenge, or at least a large part of the solution, is to do scaffold and substructure-based workflows. So how does it look like? Well, over here on the left top, we see all the structures, and okay, we see that part of them are the same, right? So immediately we start to recognize that the molecules in these complex mixtures are not independent. They share substructures, they share scaffolds. And if we are able to recognize it in the data, we can kind of structurize the data. And that is what we will do. I'm sorry, the slide is not moving on. There we go. So apologies for that. So what we see here, the structures are not independent. However, we can use metabolome mining tools to capture the similarities and the differences, and then plot it in a way that we can start to analyze the data in a more structured manner. So over here we see an example of a molecular network. I will come back to that later in my talk, and where each molecule is colored by its chemistry. This already helps us to kind of pinpoint out what kind of chemistries are in the data and where should I focus on. So as I already mentioned before, we can see that the structures of the complex mixtures are, but it also means that the fragmentation spectra derived from them are not independent of each other. So, and I think many of you know that metabolites are built from a library of building blocks. So And the word library is not chosen with that is chosen for a reason here, because libraries, you know, from books, right? Libraries have a lot of books, and books are in fact large text documents. And actually, there are parallels with collections of this text documents between metabolomics and this text documents. Why? Because in fact, the books are also not independent of each other. We have books about various topics, but they also can share topics. And that is exactly what we will use this analogy to actually find the substructure patterns. And the nice thing is that for text mining, natural language processing, a lot of unsupervised models exist. So, and that resulted in our PNAS paper on the topic modeling for untargeted substructure exploration in metabolomics back in 2016. So here you see how the analogy works going from text to molecules. Over here we have text documents, over here we have mass spectra. And on the left side, we can recognize that the LDA, latent dirichlet allocation, the algorithm finds words that often co-occur across various text documents. So all the red words here, they co-occur, right, in more than one document. And over here, the blue words, exactly the same. And the green words, again, the same. So there's two important things to note, right? So one, a document, a text document can contain multiple topics. Over here, we see two of them. And also, the same topic can occur across different documents. So here, the blue topic, blue one here, is also present over here. Yeah? There's one thing that is also important, is that the algorithm does not kind of annotate or identify the topic. We as I hope you agree that the red words are all football related and the blue words business related, etc. So, and now we're going to switch to the molecular world with the spectrums. So there's one more thing that's interesting, and that is in LDA, the number of times a word is occurring in the topic is kind of how important it is. And in the mass spec world, we translated that into the relative intensity of the fragment peak. So the higher the relative intensity, the more important the peak or the loss is for the motif. So the topics are no longer topics in our world. We term them master motifs. So basically, they are the substructure motifs that are present in the data. And again, we have more than one master motif. Okay? So in the papers, Scotland, and there was a lot of nice beers around me, ales, pale ales, IPAs. So, and of some of them I took samples, and then we measured them in the mass spec, and this resulted in as you might know, beer is a very complex mixture of yeast and plant-based molecules, and then you cook it for like an hour, so it's really full of metabolites. And over here you see a wide range of the various substructures that we could pick up using MSTLDA, but also look at how similar these aromatic substructures are, but still we were able to pick up various master motifs. In the middle here you see a network of the different motifs here, and they are connected to the blue dots, the molecules. So the larger the motif here, the more molecules were found that had this motif in them. So of course you need to validate your tool. So that's also what we did. So over here you see how we took molecules from DMPS. Back then, more than 4,000 reference standards, and mass bank, which had like more than 2,000 molecules in it, the reference set. And over here you see an example of those molecules, of this adenine containing molecules. And indeed, MSLDA was able to find a motif that is linked to this adenine core structure. And not only that, we also can recognize a green loss over here that we could also observe in other spectra. And actually we could link that to the sugar part of this adenosine molecule that we see here. And basically, this is an example of how MSLDA can decompose molecular structures into substructures and help us to annotate this. Of course, you can apply MSLDA to a lot of different samples. And Madeleine, in the next talk, will show you examples of how you can apply it to serum. Here's an example of how you can apply it to urine. So Over here, we see a lot of substructures that are typical for urine, glucuronide motif, sulfate, and a lot of amino acids, triethylamine. And also, actually, I was able to find a drug-related motif. This is losartan. It's an antihypertensive drug. I did a study in Glasgow as well to how you can use molecular networking to find drugs, to do drug screening. So that's why I was able to quite quickly identify, I annotate this motif, and this is all related to losartan, the sartan motif. That is this part of the molecule, and you can see how in 7 molecules in this urine sample, like all the 7 fragments were consistently present. So, and that's a really nice and coherent motif. So we can be quite confident that this sartan substructure is present in 7 metabolites, and that they're all likely to be related to each other. So not only that, I also found some over here. We have proline betaine, we have a fragment-based motif, as you've seen before. So these 2 fragments are consistently present in a number of molecules. Negative ionization mode data. Over here, I want to point out these are all losses. So actually, I annotated those losses, and I figured out that there were likely carboxylic acid related losses, as you can see over here. And that led me to the conclusion that this is very likely to be a citric acid related motif. And the nice thing, of course, is it links back to the motif of proline betaine, because as some of you might know, this is a marker for citrus fruit intake, and citrus away, it is citrus fruit. Joe one day put a lot of effort in building this nice MS2A.org web app. So if you have not visited yet and you're interested in substructure finding, go there and have a look around. And also you can apply for an account, and then you can start to do your own substructure finding. You would successfully have done one losses are part of it. If we fast forward 4 years to now, then we can see that we start to build this substructure ecosystem where MS2A is at the heart, and we can link the various tools to it. For example, Magma that I already introduced earlier, and also MotifDB, and those 2 I will shortly highlight now what they are and what they do. So together with Lars Ritter, we integrated the Magma tool into MS2A, and what it helps to do is to really speed up the process of substructure motif identification from library spectra. So really that's important to mention. So if we know the structure and we know the spectrum, this Magma MS2A really helps us to kind of point out what's the most likely annotation for this fragment feature. In this case, it's this alanine sub core structure. In this case, it's the sugar part is connected to the loss. And as you can imagine, in some cases for losses, it is particularly neat because they are really hard to identify using the eye. I noticed fragments are easier to find, for me at least. The losses are more tricky. And now we also, not only that, we also have an overview of the various isomeric losses that are there. In this case, you can find losses that connect to this loss of 60. And that's good to know if you want to draw any further conclusions based on the presence of this master motif of this loss. Sorry. So MotifDB is really also a very nice and important addition to MS2A. Why? It captures community knowledge. And how does it go? Well, you run your MS2A experiment, you get your motif, your master motif, and in this case it was me. You analyze the motif, you annotate the motif, and you get a ring of this actinomycin D molecule. And then I saved my annotation, this and other annotations from this Streptomyces and Salinespora dataset in MotifDB, so that next time I or somebody else having Streptomyces data can use it as part of their analysis. And that is handy because you already have a lot of annotated motifs as you throw into the mix. So, and how would it look at the moment? Well, you can run MS2A. You can capture known chemistry using the MotifDB motif sets. So if I go back, so yeah, this is an example of a motif set that you could use for positive mode data derived from Streptomyces related samples. But you also should include very short that we within the foreseeable future, we won't be able to capture all the chemistry using this motifs. So it's really important to also include 3 motifs to capture the yet unknown chemistry. Then you can start to explore and validate the known chemistry in your data. Then you can start to explore and annotate the novel chemistry in your data. And to give you some pointers there, so if you upload your data or you run your data in MS2A.org, you can go to the motif pages and explore what kind of fragments or losses are part of your motif and how consistently are they present, the combinations of these fragments and losses. And based on that, you can draw a conclusion on how nice and relevant the motif is. And for example, this looks to be very okay with more than 3-4 motifs consistently present across 7-8 molecules. So that is worth further exploring and also trying to annotate. Now, of course, I am also highly recommending you to then annotate, store these annotations in your MS2A experiment, and then also push them out to MotifDB later on so that you and others can reuse it. So towards the end of my talk, we will go over some examples. I think most of you by a really good introduction and overview of what's possible at the moment, but very briefly, and it groups them based on spectral similarity. Remember how we read the structure information earlier on? Well, here we would hope that these 2 related molecules, the spectra thereof, would be combined in one molecular family. So it can capture similar but also different chemistries. And here we see an example of a molecular network, and a molecular family would be a subnetwork thereof over here. And if we look at some of the annotated molecules, we can see that they share a lot of their chemistries. And then in that way, it did work in the sense that we were able to kind of group similar chemistries. And when we go, when we look at more complex data like this, I run a molecular network of gut extracts, and you can see the larger the dot, the more abundant it was in the data. You can see that many of the large dots don't have any library hit yet. So we're still far away from fully understanding all the chemistry in our gut. So I won't spend a lot of time on this today, but with Madeleine, Ernst, and others, we built a small and enhancer workflow. And really that is meant to capture and combine the metabolome mining tools that we have around, including MS2A, to provide both a detailed chemical look over here on the top right now. You can recognize the master motif substructures, as well as a more higher level chemical overview. And that's the part I won't cover today, but there are metabolites and the plant journal that you can read to see how that works. So, but I can highlight some results. So over here we see an example in plants where with Kyobin Kang, who's currently working in Korea, we can see how chemical overview with also kind of benchmark this approach, and where we can see how we can quickly find all the flavonoid related compounds, all the triterpenoids in the data. You can do the same for bacterial data. So again, this is data originally measured by Max Kruseman et al. And we took the same molecular network, we applied this molecular enhancer, and we start to see emerging all these different bacterial related compound classes. So if you're only interested in peptides, you can already start to zoom in on the peptidic families. Some examples to show you how then this all can help you in annotation of your data, because it's obviously the end goal of all of these tools. Increase the biochemical and chemical interpretation of your data. Over here is an example of a molecular family, no library hits, no nothing, and also the chemical annotations were all pretty low confident. However, this molecular family shares one motif, 208, and this is the corresponding fragments that I saw in MS2A.org. Again, we see consistent 2 fragments being present, and the third one also relatively consistent. And actually, based on my own expert knowledge, I could identify this as tryptophan related fragments, and hence I could start to annotate this whole family as kind of acyl tryptophans. So using the substructure knowledge combined with the molecular family, we can come really to more quickly annotation of your data. This is an example of a peptidic family of the bacteria. So actually you've seen this example a little bit before, because this is a molecular family related to actinomycin B. So all the all the purple nodes here, purple highlighted nodes, they gave an annotation using the replicator. That is an annotation to annotate peptides in your data. However, they also share, some of them share this motif 10, and this motif 10 is the motif I showed you before from MotifDB. This is the lactone ring of this actinomycin D. And over here you see the pattern presented in spectra within the msstilier.org app. Final example where we can see how from another data set from Photorhabdus and Xenorhabdus bacteria measured by Nick Tobias et al., where you can see this example of xenomycin A, which is a cyclic peptide, which has a ring and a tail. And in these families, which are related because they share the presence of a motif over here. And actually, I could annotate this motif using the replicator and FarQuest annotations of the fragments. And the blue motif here is the ring, and the red motif here corresponds to the tail. And why this is so nice? Well, actually, you can see how some nodes here only contain the tail motif, and others only seem to contain the ring motif. So that already leads us to some hypothesis on where the modifications of the peptide in this family did occur. And indeed, we can see over here, it's called red tools, and msstilier can help you to speed up your data analysis. Actually, if we apply molded enhancer to it, a lot changes because we can see much more color now, and each color is a different chemical class. I'm not going into the details today. I want to highlight that some families are gray. That key things, key challenges that I also introduced at the start of this webinar. Then this might be a way to do it, because it means that those families could be novel. To highlight some cool and novel GMPS features that I think will be really useful in LC-MS exploratory data analysis. One of them is MAST. It is analogous to BLAST for those of you that are coming from genomic data analysis. So you can take your MS-MS spectrum and you can compare it to all the public data that is available within GMPS. And for example, this is an example where I searched for a statin metabolite, and my question was, can I find it in more than one independent data set, because that would increase my confidence that this could be a real metabolite, and not an artifact just found in only my data set. And as you can see, we see it in multiple independent studies, so that increases my confidence that we have really something here, and that it's worth further exploring. Another really nice tool, MAST is spearheaded by Ming-Shu Wang, and it's already published in Nature Biotechnology. Another really nice tool is Redo, spearheaded by Alan Jarmusch and Ming-Shu Wang, and that is currently available as a preprint. And that really allows you to explore your data in the context of a lot of other data that is present in GPS. And the nice thing is that it also introduces a standardized metadata format. That is really important, and I cannot overemphasize that when you obtain your data and add it to public repositories, make sure that you add useful metadata to it so that you and others can start to use and reuse that data. So before I wrap up, I just want to highlight that the current project that I'm leading, and with whom I'm collaborating with the eScience engineers from the Netherlands eScience Center, is really working at the various levels that I discussed. So at the data level, we look to see, can we find combined genomic and metabolomic data sets. So we set up the paired data platform, paired omics data platform, as we call it nowadays. Google it and you will find it. Have a look. If you have data from bacteria which you know are sequenced, please add it. We are mainly looking for LC-MS data sets at the moment that we can link the metabolomic data to the genomes. Structures, well, I already showed you examples of how we can use annotation tools to increase structural knowledge of the data sets. And of course, I don't need to introduce the substructure part of the project, because that's what I showed today. So with that, I came to the end of my talk. I really want to thank you all. I hope I showed you how the metabolome mining tools combined with the annotation tools that are all available these days can really help you to speed up your metabolomics analysis. If you're interested in my work, you can find me on Twitter. You can also find me on LinkedIn, Google Scholar, and ORGIT. And with that, I want to thank you for your attention, and I give the floor to Madeleine.
Thank you very much.
Yes.
So first of all, good afternoon, and many thanks for inviting me to this. I'll introduce you to some of the metabolomics work we do at Statens Serum Institute here in Denmark. And as my presentation time is only very short today, I decided to give you a broad overview of what kind of research we do and how we use some of the metabolome mining tools just introduced to you to address questions within the clinical metabolomics field. The public health areas of disease in newborns, and more recently also the COVID-19 test center. Our institute also hosts the Danish National Biobank, which with 8.5 million biological samples in store is one of the biggest and most advanced biobanks in the world, offering very unique opportunities for clinical research. And so I myself, I'm based at the section for clinical mass spectrometry at the Danish Center for various inborn errors of metabolism. Afterwards, all of these dried blood spots are stored in the National Biobank, and they currently sum up to about 2 million samples, which corresponds to about half of the Danish population. And so because of the very unique Danish system, where each person at birth receives a unique identifier number, we can then link these dried blood spots to diverse Danish registries and, for example, retrieve information on later development of diseases. Sorry. And so in this context, these samples provide a very unique opportunity for us and our many collaborators to address diverse research questions within the clinical metabolomics field and related to neonatal health. So we use these samples to, for example, investigate whether there are metabolic predictors of childhood asthma at birth, or whether there are metabolic anomalies at birth that can predispose to mental disorders. But also questions such as how does the metabolome of naturally born children differ from C-section born children, or what defines the metabolome of extremely premature children. And so to address these and other questions, we submit our samples to untargeted metabolomics workflow, which usually looks something like this. So we prepare extracts of our dried blood spots, submit them to tandem mass spectrometry data acquisition in DDA mode, and then use several of the data mining tools just introduced to unravel biochemical underpinnings of neonatal health and biological hypothesis creation. And so just to give you one example of our studies, so in one of our studies, we tried to investigate how the time spent in uterus influences the neonatal metabolome, or in other words, how does the blood metabolome change with gestational age? And so why does this matter? So prematurity, which is defined as a baby born before 37 weeks of gestation, is a very complex and severe pathophysiological condition, which along with a number of short-term consequences such as bacterial infections also leads to an increased risk of several late-term adverse health conditions such as cardiovascular disease, diabetes, or a number of very severe mental disorders. And so to investigate how the neonatal metabolome changes with gestational age, we sampled approximately 150 samples from the National Biobank from babies born at different levels of prematurity. And what we basically could see is that a large variation in the blood metabolome of these newborns can be explained by gestational age, which is here illustrated with a principal coordinates plot. So we can see this of the variation data with a relative. So the information we had at this point is a large variation in the metabolome can be explained by gestational age. So many metabolites would either correlate negatively or positively with gestational age. However, all of these molecules at this point were unidentified. And so this ever-increasing accumulation of unidentified metabolites is actually a major bottleneck in clinical metabolomics studies, which have logical interpretation and hypothesis generation. The MolNet and discover potential and potential discovery of novel biomarkers of disease, of finding truly novel biochemical underpinnings of disease. And so what we could, for example, see in this study is that molecular families carnitines, nucleotides, and polyamines were significantly related to gestational age. We also found some metabolites related to gestational age that are related to the care or the treatment that is given to these very premature babies. Now, while this finding is probably not the most exciting from a biological point of view, it still gives us a good confirmation that our methods actually work and that we are able to pinpoint metabolites that are in one way or another related to gestational age and prematurity. Of course, we also found some metabolites for which we did not find any chemical structural information, but as Justin highlighted, for these metabolites, we could use the novel MAST search tool on GNPS, and we could observe that some of these metabolites were previously found in blood, fecal, and skin samples, as well as some bacterial isolates. So leading to, although very vague, hypothesis that maybe these metabolites could be of microbial origin or antimicrobial origin. On this note, we also found amino acid-derived compounds that were significantly related to gestational age, and some of those were previously described to be modulated by bacteria in the gut. And some of the metabolites that are highlighted in blue here that were more predominantly abundant in very premature babies were previously also associated to diseases that can be late consequences of preterm birth, such as cardiovascular disease, diabetes, and schizophrenia or cognitive impairment. And so summarizing, this study allowed us to pinpoint several metabolites that are significantly related to gestational age or to prematurity, and allowed us to create a hypothesis that we now can test in future more elaborate studies. So basically, we found that many metabolites that were correlated with gestational age were related to treatment, such as antibiotics. Then we saw some metabolites that are eventually related to the diet, such as nucleotides. So many prematurely born children are fed parenterally. And we also found polyamines, which are related to cell growth, so maybe gut maturation, but also metabolites that are known to be modulated by the gut microbiome, such as bile acid carnitines and amino acid-derived compounds. And some of these metabolites have also been related to adverse health outcomes that could be related to premature birth. And so it is known that antibiotics use as well as diet has a large impact on the gut microbiome. And the gut microbiome, on the other hand, is known to have a profound impact on health, especially very early on in life. And so as we can observe all of these metabolites in our dried neonatal blood spots, a very nice outcome of our study, although of course in a very more distant future, could be if we could use these dried blood spots to screen for those metabolites, and with that, for example, inform probiotic or dietary intervention to eventually prevent short as well as long-term health risks related to preterm birth. And so with that, I'd like to thank you for your attention, and of course, a big thanks also to all our collaborators and the funding agencies. And yes, that's it from my side. Thank you.
Great. Thank you very much, Madeleine, and thank you very much, Justin, for your great talk. We received some questions and even some comments saying nice talk. The first question that we received for Justin. When you say the fragments are not independent, can you further explain? Are they not independent because different metabolites will belong to different classes of molecule? For example, all phospholipids have to have a phosphate motif. Justin, could you please answer to that question?
Yes, I'll try. So what I meant with that is that if we fragment metabolites and if we fragment structure-related molecules, then the assumption is, and I think this I have shown in my talk that this is true in many instances, that structure-related or very similar molecules produce related or very similar fragments. For example, glycosylated molecules often share losses related to the glycosylation molecules contained. So in that sense, the fragments also not in that was fragmented, but together they are derived from a substructure or scaffold.
Do Elena, do you want to take the other question?
Up to you. It's
yeah, I can go ahead. So we have another question saying to you, for you, Justin, saying you have been talking about losses in the structure. Can it be possible in untargeted metabolomics where you can have losses with similar Dalton? Yeah.
That's a question?
Yeah, that's a question.
Okay. Yeah. So yes, and actually I showed an example of that in when I showed the magma MS2A integration, where one loss of 60.02 something had like at least two isomeric structures in there, right? So the configuration of the carbon and oxygen and nitrogen atoms was slightly different. So yes, it's possible, and that is something to take into account when analyzing the data.
Another question for you: is it possible to use MS2 LDA with MSC data?
With what kind of data?
With MS2DE data.
So in theory, yes, as long as you produce data in let's say MGF format, where you have the precursor ion and fragments, right? So for example, I know that for a Q executive instrument, you can use this step collision energy function that I also used when I was in Glasgow, when I actually still measured my own data. So, and that also, it's not MS2DE per se, but it also combines various collision energies into one spectrum.
Right. Another question for you: how about novel structures or structure which are not in a library or classifier?
So that's okay. So there's a few things to say about that. So structures as a whole are quite often not present in the database, right? So typically, for really complex mixture, 2 to 5% you will find a hit in the library, and that's not said that they're all correct as hits. So be careful with that as well. So the chance is large that the structure is not there. However, substructures, you already increase your chance a little bit, because as I repeatedly said during the talk and tried to show, and the metabolites observed are not independent. They share scaffolds, they can share substructures. So however, I think still we don't know a lot of the substructures or haven't annotated them yet. So yes, in that sense, you can start to try to annotate those substructure patterns or master motifs by using fragment searches in Madeleine, MZ-Cloud, MassBank, and you name it, to try to see what kind of structure could relate to this particular mass fragment feature, and then you do it for another one in the motif and another one, and then a pattern may emerge, right? If they all point towards a similar substructure in various spectra, then you can start to form a hypothesis that the motif is actually pointing to that particular substructure. Does that always work? No, but at least it could be a way forward. And if that doesn't work, you could always try to isolate one of the molecules in your data that is most abundant and do the usual kind of more in-depth stuff, NMR, to try to elucidate the structure. And after you are elucidated the structure, you can then, of course, annotate the master motif and add the motif to MotifDB.
Right. I just had a look at the time. Is that we are a bit over? Is that okay for you to stay a bit longer to answer to the numerous questions that we did receive for you too? Yeah, that's fine.
No.
Yes.
So I have another question for you, Justin. Do we specify before running the analysis whether we use positive or negative mode?
The very short answer is no, in the sense that the algorithm doesn't really matter what you put in, of course. For you, it matters, right? So I would definitely put it in the title, so you know this was positive mode, this was negative mode. And for the use of MotifDB, motif sets, it matters because some were obtained in positive mode ionization and some were obtained in negative mode ionization. And so it's very important that if you want to use those motif sets, then check that you use the right motif sets derived with the right mode, but also from similar samples or from reference compounds that you think may be relevant for your study, right? If you have urine data in positive mode, then I know that there is at least one motif set that could be relevant for you. But if you have fecal data in negative mode, probably none of the current motif sets are relevant for you. So I hope in the future, of course, it will change, and we have many more motif sets from various ionization modes and sample backgrounds, so that slowly we can start to capture the complete chemistry.
Great. So someone who actually joined me to say thank you for the very nice talk. I'm wondering how you deal with MS2 purity and the fragmentation of collating species. And also, did you run your sample at different collision energy, or is the experimental setup in untargeted metabolomics project?
Question to whom?
Sorry?
To whom is the question?
Oh,
I mean, Madeleine can also
to you, Justin.
So as I think as you've seen now, I work in Wageningen at the bioinformatics lab, and there we don't have like a wet lab. So at the moment, I don't really run my own samples. So as you have seen during the talk, I've been able to reuse public data from GMPS for the method development, and also as to showcase how this new mining tools can work. However, when I was in Glasgow, I was still measuring my own data, and there I've been mainly working on thermal instruments, and they have this step collision energy. So there I could combine the various collision energies, and also I've worked with multiple scans in 30, 60, and 90 normalized collision energy, for example. So, and of course, it's important to consider the collision energy because it is very dependent on the collision energy, how many fragments you see, and just how much structural information you can derive from the metabolite feature. And I think that only partly answers the question. So what was the other part of the question?
So the other part of the question, how is also did you run your sample at different collision energy? But you just already answered that. And how is the experimental?
I remember the chimeric spectra. So this is a problem that, or this is a challenge issue problem that, of course, MSLDA or molecular networking on themselves cannot solve or cannot easily solve, because they do with the data that comes in. However, if you think about ways to overcome that, at least in part, is to change your chromatography a little bit to get longer chromatograms. If you really have very complex samples that contain a lot of isobars and stuff like that. But of course, that is at the cost of longer measurement time, so you lose a bit of your higher throughput aspect of the metabolomics workflow there.
Mm-hmm. We have still a question for Justin, but I might address some for Madeleine, if you don't mind, Justin.
Oh, sure, go ahead.
Thank you. So the first question to Madeleine was, I'm wondering why the choice of DDA when DIA can capture more metabolites, which may represent a truer picture of the neonatal metabolites. So it's basically why DDA rather than DIA. Madeleine, we can hear you.
Sorry. So our unit is basically relatively new in the field. So I joined the group just a year ago. And basically this is all data that has already been acquired. So it just was the way it was done before. Mm-hmm. But we aim at acquiring new instruments and experimenting as well with different types of mass spectrometry data. So that was just an example.
Right. Another question for you, Madeleine. What methodology did you follow for the calculation of metabolite predicted gestational age?
So basically, you can refer to the preprint we published, but we use different methods. So on one side, we built a lasso model to try to predict gestational age based on the metabolic features we detected. We also did univariate correlation, so to assess the correlation of individual metabolites with gestational age. And thirdly, we also, in collaboration with Dr. Simon Rogers, tried to identify molecular families that are overrepresented in metabolites that correlate with gestational age.
Right. I saw another question for you, Madeleine. Or how metabolites link to a mechanism that explains the relationship between the metabolite and the disease?
And sorry, a metabolite and?
How metabolite linked to a mechanism that explains the relationship between the metabolite and the disease?
And sorry, I don't get the last word.
The mechanism. So maybe it's my French pronunciation, I'm sorry. Can you see, maybe you can see the question in the mechanism? Mechanism. Thank you, Justin.
Okay, so the relation of the metabolite with?
The mechanism between, which explains the relationship between a metabolite and the disease. How are the metabolites?
Okay,
yeah.
Metabolite with, okay. Yes, so this is the ultimate idea we want to do. The data I showed is just a very preliminary pilot study. Mm-hmm. So we didn't actually have access to data about disease for these samples because this has to be retrieved from the Danish registries. Right. But this is definitely for follow-up studies.
Great. I'm going to take another question for you, Justin. What are the challenges to integrate many results from different programs?
Well, that's a very broad question, but so for example, if you think about output formats, right? So many tools have slightly different output formats, and then when you want to combine those outputs in one place, you need to somehow bring those outputs back together. I think what we can observe is that there start to emerge various ecosystems. So around, let's say, GMPS, molecular networking, around MS-Dial, and around MS-Mine, where multiple parts of the pipeline are now integrated, and then of course this helps also because the output then is also integrated. And today I showed you this molecular networking output. So inside the scape, you have then all the information combined, which means that you can more quickly see if you can correlate things you find, let's say, from the annotation software with the mining software.
Right. Okay, I'm going to keep going for a little while. There is many questions. You stop me if you need to go. I'm going back to Madeleine. There is 2 questions for you. So from the same person, what is the advantage of storing samples as blood spots versus frozen liquids? And how well does extraction work? And he's very interested into your biomarkers for psychiatric disorders.
Okay, so I'm focused on the data analysis part, so I don't know incredibly much about the experimental part. But basically, we are focusing on dried blood spots because these are the samples that are stored in the National Biobank and from which we have almost half of the population of Denmark covered. So we really aim at investigating research questions with these samples. Now, how stable these samples are, this is just questions that are started to being investigated and that we also will address in future studies.
Right. And there is another question for you. Does the Danish system for blood sample collection extend to the full lifespan of the Danish community, or is it limited to newborns?
So the biobank hosts different tissue types, and you can go to the website of the biobank and like do a search of what kind of samples there are contained. But so the dried blood spots, they make up a large majority of the samples.
Right. And do you have access to fecal samples of newborns? Would it be possible to link the metabolite in the blood to the gut microbiome diversity of baby, for example?
Yeah, this would be a really interesting study. There we would be relying on collaboration. So it's not our own group that does that. We're just focusing on the metabolomics part.
Right. Okay, I'm going to do 1 or 2 last questions quickly. There is still some question, but I guess the speaker will be happy for you that you contact them as they put the social media contact on their talk. So for Justin, can the workflow be used with GCMS spectra?
Sorry, can you repeat the question?
Can these workflow tools be used with GCMS spectra?
The GCMS spectra.
Yeah.
So actually, at the moment, we have not implemented that, but in theory, they could, right? If you look at GCMS spectra, also there, the same principle holds that fragments, the spectra fragments derived from similar structures, they often share, are similar, or they share the same patterns. So in that sense, the answer is yes, but at the moment, it's not implemented yet. For example, a key difference is that in LCMS, we normally have a good idea of the parent ion, so we can also calculate loss, determine losses, and use those. In GCMS, this is normally not the case. And also, if you look at the accuracy of the data, most GCMS instruments have nominal mass, so this also will impact how you can use the pattern mining. But it's certainly interesting if people are interested to set up a project to kind of explore that and initiate that, then please contact me and we can see what we can do.
Great. Well, I think we're gonna start.
Maybe I can also add that recently GCMS analysis is possible within GMPS, so that's also a nice way to start to do some LC-MS exploratory data analysis there. So have a look, and maybe that already helps you a little bit on the way. For example, the molded enhancer workflow has been adapted to also accommodate GCMS data. So
Right. Well, I think everyone is joining me to thank you, and as well as to thank the EMN for organizing the webinar. So I enjoyed this opportunity to say that the slides are this webinar have been recorded and it will be uploaded on the Metabolomic Society website. So you will be able to access the slides and the explanation as well as the question. And don't forget to check our next webinar. So yes, with that, I would like to thank you again, Justine and Madeleine. We were thrilled to receive you for the EMN, and I wish you all a very good weekend and stay safe.
Thank you, Marine. Thank you very much. Thank you to the EMN. It's great to see you so active still, and please all go to the next webinar as well. Purva is also does very nice research, and please join for her webinar again. So have a nice weekend.
Thank you.
Thanks for joining, organizing.
Thank you very much. Bye.
Bye.